With more than 6,000 small and micro-cap companies listed, if you're looking for the next Apple at the earliest stage, then Channel Check truly is the orchard. The listed companies support Channel Check, so it's free for you, the potential investor, to gain access to institutional quality research from FINRA licensed analysts, advanced market data, industry reports, news, and a growing catalog of videos and webcasts. Make sure to subscribe to our YouTube channel and register for Channel Check so you're always up to date on what's going on at the small and micro-cap data place. Welcome to this special edition of the C-Suite series presented by Channel Check and Noble Capital Markets. Noble is an SEC-registered FINRA-licensed broker-dealer and the source of the equity research available on Channel Check. This investor forum at the World Stem Cell Summit presentation features ImmCelz, a subsidiary company of Creative Medical Technologies Holdings, OTC ticker symbol CELZ. Following a brief overview presentation from Chief Scientific Officer Dr. Thomas Eeckham, Noble Senior Research Analyst Robert LeBoyer will moderate a Q&A session. If you registered for this event through channelcheck.com, in the coming days you'll be given an opportunity to get your own questions answered by the company. With that, I am pleased to present Dr. Thomas Eeckham.
I would like to begin by thanking the organizers for inviting me to speak in such a distinguished forum. My name is Thomas Eeckham. I am the Chief Scientific Officer of Creative Medical Technology Holdings and of the subsidiary called ImmCelz, which is an immunotherapy company. Because our parent company is a public company, this is a typical forward-looking statements, safe harbor. So I'm going to begin by telling you a little bit about our parent company and the history, corporate history. We were founded in 2011. We ended up publicly trading in 2016 under the ticker symbol CELZ. The basis of our company was somewhat unorthodox. We wanted to be a biotech company with a cash flow. And to facilitate this, we began by developing autologous point-of-care products, if you will. The first one being CaverStem, which is past clinical trial and is commercialized now. It's for a urological indication. And other ones in the pipeline include FemCelz, which is for female sexual dysfunction, a regenerative approach, StemSpine, as the name indicates, for lower back pain, and OverStem, which is utilizing a regenerative approach to ovarian dysfunction and ovarian failure. What we're going to be focusing on today is the subsidiary, wholly owned subsidiary called ImmCelz, which is a very interesting combination between an autologous and an allogenic product. It's a first-in-class therapy, which I think you'll find very interesting. So a little bit about ImmCelz. We are a wholly owned subsidiary of Creative Medical Technology Holdings, ticker symbol CELZ. And our product, we call it a first-in-class therapeutic because we leverage immunotherapy to induce regeneration. It's a very new and interesting concept which has been around, if you look scientifically, but it's never been commercialized, utilizing immune cells to stimulate regenerative processes. And we like to call this not autologous, not allogenic, but allogenically influenced. Essentially, we extract immune cells. In general, and the main product is called peripheral blood mononuclear cells, cells from the leukopak. We extract these cells from the patient being treated. We expose them to standardized allogeneic cell population, and then we infuse the patient's own cells back after they have been exposed. This is why we call them allogenically influenced or ex vivo reprogrammed. So to understand how our therapy works, it's important to say a few words about the concept of how stem cells work in general. The original concept of stem cells, of adult stem cells, was that when the tissue is injured, the stem cell homes to the tissue and differentiates. And this was a very easy idea to understand. The idea that the bone marrow might become cardiac cells, hepatic cells, liver, kidney cells, and so on. Unfortunately, these experiments, the initial experiments, which were in animals, where they would label stem cells green, GFP stem cells, then they infuse them in a mouse, then they give the mouse a heart attack, and then the mice you saw beautiful green, implying that the bone marrow differentiated into cardiomyocytes. Unfortunately, a lot of those experiments were not reproducible. When you looked at humans that were treated, females treated with male cells, the male cells did not differentiate into heart or into liver. So that caused, even though the patients were having improvement, so that caused for a reevaluation of this idea that the bone marrow and that adult stem cells differentiate into tissue. See on this figure on the upper right, that's a differentiation hypothesis. And that ended up shifting into the growth factor trophic hypothesis that when you infuse different stem cells, the stem cells are stimulating endogenous regenerative processes. The stem cells themselves are not regenerating, but they're providing growth factors. So we took that concept a step further and we said the stem cells are endowing immune cells with regenerative abilities. And we can discuss this in more detail in the Q&A. But you know, one example of regeneration, immunologically induced regeneration, can be seen in pregnancy. During the formation of the placenta, the maternal T cells are actually important in stimulating new blood vessels. If the maternal T cells are not recognizing the fetal antigens on the trophoblast, then you have non-functional formation of placenta and spontaneous abortion. So there's a couple of other examples. So what we tried to do is we tried to stimulate regeneration using immune cells that have been programmed by stem cells. So what supports for our work, you know, when people inject stem cells intravenously, they go in the lung, the majority of them. From the lung, they secrete growth factors. The strongest evidence of stem cells inducing an immune modulation, a systemic change, is one of the publications on the right-hand side of the slide, in which ejection fraction, measured as fractional shortening of the heart, increased in an animal model of heart failure, of myocardial infarct. The ejection fraction increased when you inject stem cells, not just in the heart, but when you inject them in the thigh muscle. So the stem cells can mediate systemic regenerative activity, and in some of the work we've done and work that other people have published, it's been shown that stem cells induce generation of T regulatory cells, and in some cases the T regulatory cells induce regeneration. So what we did is we sought to optimize this. So we, in the ImmCelz product, we take universal donor cells, which are cultures with the patient's own immune cells, who are conditioned by exposure to factors made up by the immune cells. We reprogram the patient's own immune cells, and then we put them back into the patient. Here is a pictorial example. Basically, number one, you extract the patient's cells using leukapheresis. It's the same technology that's used for CAR T cell generation. You incubate them with either through transwells, so there's no contact, or you incubate them with conditioned media of universal donor stem cells. When you do this incubation, you also have the ability, and we've done this through different versions of our product, where we culture with other agents such as low-dose interleukin-2 or rapamycin to enhance the reprogramming. Then we go through a quality control system check, and then we infuse back in the patient. Basically, this is similar to what Steve Rosenberg was doing back in the late 80s, where they were doing tumor-infiltrating lymphocytes, and to what's being done now with things such as CAR T cells, where you take out the cells, you manipulate them ex vivo and put them back in. The only difference is the regulatory burden here is a lot less, because the manipulation is not a genetic alteration. The manipulation is an exposure to stem cells in a contact-independent manner. So let's talk about some of the data we have. We've been able to show in vitro, there's some of the basic properties, immune modulations at an innate level of the immune system, immune modulations at the adaptive level, stimulation of androgenic activity by the immune cells, and induction of regeneration. And remember, you would like to induce regeneration with immune cells because immune cells, they home a lot more in the body than stem cells. For example, a T cell is about half the size, quarter to half the size of a mesenchymal cell. Mesenchymal cells, if you infuse too many of them, they get stuck in the lung. T cells can travel, and they do travel all over the body. So let's take a look really quickly at some of the data. This is production of TNF-alpha. It's a mediator of innate immunity. We have data like this with other innate immune cytokines. So if you look at 12 hours, the blue is control, and on the y-axis is production of TNF-alpha inflammation. So when you stimulate with LPS, you see inflammation in the orange and the gray. When you give the PBMCs, which is the blood cells from the patient, when you give them before conditioning, before treating with the stem cells outside of the body, the gray, you see they do not do much. When after you expose them to the stem cells, they're reprogrammed. You can see the yellow at 12, 24, and 36 hours, potent suppression of TNF. In this figure, we look at interleukin 17, which is a marker of adaptive immunity, and we also see a potent suppression of interleukin 17 production. In this example, we take the cells, patient cells treated with stem cells or without stem cells, and we incubate them with something called human umbilical vein endothelial cells. This is a marker of angiogenesis, production of new blood vessels. You cannot have tissue remodeling, you cannot have tissue healing without production of new blood vessels. And here we demonstrate with increasing ratios. So on the left-hand side, you see 0 ImmCelz to 1 HuVec cell. There's no difference. When you do 1-to-1, you can see an increase in proliferation. When you do 1-to-2, you see a higher increase. You see the same trend when you look at VEGF production, which is a regenerative cytokine also associated with angiogenesis. But does this work in vivo? Those were in vitro data. We believe it works in vivo by inducing an immunological phenomenon called the tolerogenic loop. Essentially, here is you're inducing generation of immature dendritic cells. The mature dendritic cells then go stimulate production of T regulatory cells. Then the T regulatory cells go and make more immature dendritic cells. Basically, induction of self-tolerance, which is an active immunological process. And so, our first evidence of this is in the collagen-induced arthritis model, where you inject female mice with type 2 collagen, and then there's an immune response. This is the days after the second administration of the collagen. And on the y-axis, you can see is a clinical arthritis score, how bad the arthritis is in the animals. You can see with the X's, once you give the ImmCelz to these animals, the disease goes up and then it goes into remission. Interestingly, when you deplete T regulatory cells, there is no therapeutic effect, implying that there is an immune modulation that's T regulatory cell dependent. Similarly here, when you look at tolerogenic dendritic cells, you see a reduction in the number of tolerogenic dendritic cells when you take away T regulatory cells, implying in this in vivo example, we are inducing tolerance through induction of this interaction between T regulatory cells and tolerogenic dendritic cells. Now, does this work in other models and in models of bona fide degeneration? And rheumatoid arthritis, as we know, it's degeneration, but it's immunologically mediated. So here is one of our programs, which is liver failure. Liver failure is a significant cause of morbidity and mortality. Obviously, there was a reduction in liver failure with the work of Gilead in curing hepatitis C. However, it's still a significant problem internationally and in the USA. And as many of you know, the main cause is cirrhosis, which is activation of the stellate cells in the liver, causing scar tissue. It's associated to some extent with immunological processes. We thought, however, if you give, for example, cyclosporin, it doesn't help in liver failure. So there is obviously a more insidious degenerative element. We used a commonly known model called the carbon tetrachloride administration model. Here are some of the data. This is on the left-hand side is ALT, and you can see administration of the cells. So if you look at the lines, the first one is, if you look at the 24-hour, the first one is control, no carbon tetrachloride. the one that's darker, carbon tetrachloride. The one that's higher than that, carbon tetrachloride plus cells, T cells that have not been trained, that are not made into ImmCelz. Interestingly, T cells that are non-modified seem to make the phenomenon worse, to make the pathology worse. When you train the T cells with the stem cells to make the ImmCelz, you actually see a much profound reduction throughout the time points. The other program we're working on, and Dr. Camilo Ricordi from the Diabetes Research Institute recently joined our scientific advisory board, as type 1 diabetes, which, as you know, is a significant cause of morbidity and mortality, one of top causes of blindness. Transplantation is the only cure. Obviously, it's immunologically mediated. The problem is with transplantation, you need multiple donors. You need multiple donors, and there are not enough donors, obviously, for recipients. And even when you do the transplantation, you are not curing. Right? You need to put these kids on lifelong immune suppression, which comes with its own risks. So here is the ImmCelz in the accelerated model of diabetes. You can see at the day 7, there's some effect. Day 14, more profound effect. Day 21, very profound effect of the ImmCelz in terms of reducing the diabetic incidence. This is associated with increases in EGF, epidermal growth factor, locally in the pancreas, insulin-like growth factor in the pancreas, hepatocyte growth factor in the pancreas, which is very interesting, suggesting there is an immunological and a regenerative phenomenon going on. The other interesting thing in our work with type 1 diabetes is that it seems to synergize with other therapies, such as BCG, in reducing the incidence of diabetes. This suggests ImmCelz might be a powerful adjuvant, especially given that there's different approaches that have been tried and are being tried for type 1 diabetes. You know, these kids have a honeymoon period, and for example, GAD65 peptides have gone through phase 2 and even phase 3 trials. There was some level of efficacy, but not enough to get them registered. We believe a lot of drugs can be resurrected by combination, and of course, we're also looking at the possibility of using ImmCelz in different combinations. Also, it increases GCSF activity. So the scientific conclusions here are that ImmCelz is a regenerative immunotherapy. That's the way we look at it. The first regenerative immunotherapy product in which immune cells are endowed with ability to induce stem cell-like activity. It's personalized, which we believe provides an increased element of safety. And right now we have several mechanisms of actions that are elucidated. So we believe strongly, not just in seeing the data, that it's therapeutically efficacious in the animal models. We're also we have our heads around some of the main mechanisms of how it works. And from the commercialization, here's where we're at a biotech conference, so we need to say something about the commercialization. We have an IND number issued. We are going for a 32-patient clinical trial, 3 doses of cells, 1 placebo, 8 patients per group. This is a 6-month study that we proposed to the FDA. We want to see safety with signals of efficacy in patients 7 to 30 days after the onset of stroke. For efficacy, we're looking at the standard markers, Rankin scale, NIHS scale, a Barthel scale, and for toxicity, we're using the WHO toxicity criteria. So thank you for listening to the presentation, and I'm honored to address any questions you have.
Thank you, Dr. Eeckham. I have a few questions for you. And the first one is, how did the idea of using immune cells as a substitute for stem cells come about?
Thank you. That's a very good question. So the idea always has been that stem cells mediate their activities in different parts of the body than the specific place where they're injected. So there was a very groundbreaking study in guinea pigs in which stem cells were injected into the thigh muscle, but they were having systemic effects, for example, in the heart failure. So the idea was, first question was, how are they doing this? And as we discussed in the presentation, the initial thought was that stem cells hope But when you put stem cells in a muscle, they cannot hold. They're pretty much stuck there. And the amount of stem cells that leak out are so small. And obviously, when you check in the hurt muscle, you do not find it. So the idea was that there must have been some kind of mechanism at play. And the mechanism of the stem cells secreting growth factors, people always believed in. But the problem is some of the therapeutic activities of stem cells are long-lasting, of allogeneically implanted stem cells. So that always made us to suspect that maybe the stem cells are modulating some of the immunological process. Now, can immune cells actually act as regenerative cells? And in the beginning, you would think no. But as you look, one interesting example we mentioned already is pregnancy, but the other example is cancer. There are studies in cancer. We like to think that the immune system's job is to suppress cancer, which in many cases it is. But if there's many animal studies where if you take animals and you deplete them of T cells and you give them cancer, the cancer actually grows a lot slower. And this is because in a lot of animal models, at the beginning phases, the cancer programs the T cell to have the T cell make growth factors that stimulates the cancer to grow. So we have examples of T cells secreting growth factors like hepatocyte growth factor in cancer. We have examples of T cells secreting things like placental growth factor in pregnancy. So we asked ourselves, maybe stem cells can modulate T cells. Now this is, you may notice in the presentation, we mentioned ImmCelz and we mentioned immune cells. I do not want you to think that we're only stuck on T cells, because there's actually much profound changes that happen to the monocytes as well. So to give you the short answer, we believe that the alterations that the stem cells do to the immune cells, we just came across that idea by following nature, following what happens naturally in the body and trying to recapitulate it.
Yeah, you touched on some of the points you made during the presentation, which go to the origin of the product. And do you believe that it's one immune population in ImmCelz that's mediating the therapeutic effect, or more complex than that?
Well, it's very interesting because as biotechnology drug developers, we always want to have a magic bullet, if you will. Unfortunately, the more and more we study cellular therapies, we realize that there's always a symphony of cytokines going on, if you will, and a symphony of activities. Some of the people at the conference may remember the company Astrum, and Astrum had this very nice bioreactor where they were incubating bone marrow cells. And of course, the main mechanism of action is the mesenchymal cells, but as the company found out, it was the interaction between the mesenchymal with the type 2 monocytes, with the T regulatory cells, that they were growing. Similarly, I believe in the first generation of the ImmCelz product, where we're doing peripheral blood mononuclear cells, we believe that it is an interaction that is happening as we are growing, as we're culturing ex vivo, and it is a relatively short culture period. But as we are growing this ex vivo, the stem cells with the peripheral blood mononuclear cells, we believe, and not just we believe, we have some data where we show that if you take out certain components, you lose activity. So I believe it is not a one cell phenomenon, but it is a multiple cells working together in synergy that are going on. That is an area of very active investigation by us right now, because we want to identify what are the molecular signals and to what extent can some of this be recapitulated artificially without having the need to use the patient's own cells. So it's very exciting times for us at ImmCelz right now.
Yes, certainly very exciting times in the field. And as you pointed out, it's very rare to find a single point of action that acts like a silver bullet. It's more likely to be a pathway and coordinated processes, as you described. So that brings up the idea of barriers to commercialization for a personalized autologous product. And what do you foresee in commercialization between where you are and the market?
Well, thank you. This is very, very important question because, you know, if you were to ask this question about 5 years ago, everybody'd say everything has to be allogenic. You know, because allogenic, it's a standardized model where everything is made from one cell, from one batch, and personalized medicine, personalized cellular therapy always had the issue that every drug dose is an individualized dose. But what we've seen with the commercialization of the CAR-T therapies, if something works, people find a way around it. And I think people who have commercialized CAR-T therapies have really forged the road ahead for us and made things a lot easier, at least in the minds of the physicians and the users of that product, where the concept now of just taking the leukopacks, shipping them to a point of processing where they get processed and then being shipped back, now is not as outrageous of a proposition as it was maybe 5 to 10 years ago. I mean, you have to remember, back even in the late 80s, early 90s, Steven Rosenberg at the NIH, he was doing something very similar. He was taking tumor biopsies, dissociating the tumor to get out lymphocytes, going and expanding them outside of the body, and then infusing them back in. You know, companies such as Iovance now, I mean, they have a similar business model. So I think it's becoming more and more accepted, the idea of personalized medicine and of personalized cell therapy. I think that at the end of the day, everybody's body is unique. And
Yes.
sometimes it's good to augment the body's own regenerative or therapeutic activities and let the body decide what it needs. And the purpose of the intervention is just giving it a hand, as opposed to coming and flooding the body with something that it may or may not need. So to answer your question directly, there are obviously potential barriers. You know, it's a lot easier to have one cellular dose, a one size fit all, a universal donor cell, if you will. But on the other hand, I think sometimes we sacrifice the convenience for potentially higher efficacy.
And speaking of efficacy, the other side of the coin would be side effects. What would be the potential adverse events of ImmCelz?
Well, so here's another interesting question. These are autologous, but I have seen so many times people saying just because something's autologous, it's safe. And that's very preposterous. Whenever you take something outside of the body and you manipulate it, there's always a possibility of something happening to make it not safe. I mean, people say that naturopathic medicine is safe because it comes from plants. The natural, well, you know, belladonna is also a plant, and you can't be eating belladonna. So the thing is, with the autologous therapies, we have not seen any adverse effects, but we also, in the processes that we submitted to the FDA, and as we're scaling up for the clinical trial, you have to be very careful. You obviously have to be careful anytime you do a cell therapy with things such as endotoxin contamination, mycoplasma contamination, bacterial contamination. Our ex vivo culture is relatively quick, so it's not one of those 14, 30-day cultures that you do with other cells where you always have possibility of contamination. Well, there's always possibility of contamination, but when you have a culture more than a week, those possibilities of contamination end up going exponentially higher. And there's also contamination possibilities by things that we are not fully aware of. Remember, the things that we test for as an industry are contamination sources that have been known for a long time. But as you may remember from the whole discovery of the gut microbiome, there's 99% of things in the gut we cannot detect using standard methods. You have to use the as ribosomal-less sequencing. Same with when you're culturing cells. When you do a long-term culture, there's always possibility of contaminating with different things that we're not even testing for. So there's always possibilities of risk. The fact that we have a short-term culture, I think substantially decreases our risk. I also think the exciting thing is that we have not seen any adverse effects because the cells are autologous cells that are only conditioned, and when they're conditioned, they do not go cell-to-cell contact with the mesenchymal cells. So the mesenchymal cells secrete different products, they reprogram them, but they do not permanently change the cells. So we believe that in the scale of things, this is not, it's not something like, for example, in CAR-T, where you're permanently ablating the B cell population of people. And the thing is, everything is about risk and reward and so on. But I'm saying, out of the scale of invasiveness of interventions, this is probably one of the least invasive cellular therapies, based on what we know.
Yes, that's a very good point about the invasiveness. And would you say that this is a first-in-class therapy?
Well, that's what we are looking at it as. And the reason why that is because, to our knowledge, now of course, maybe other companies, we've checked extensively, and our patent lawyers obviously have checked, and our medical advisors. But to our knowledge, we're the only company that actually is doing this allogenically influenced, if you will, approach, where we take the autologous cells, we let them play around in the tissue culture without actually touching the stem cells, and then we put the autologous cells back in. So we believe that this is a first-in-class therapy, and we believe, our intellectual property portfolio is very, very strong. We're filing numerous patents to protect all sorts of aspects. But as with any exciting therapy, we believe that other people will come in and start trying maybe different variations that we did not think of. And obviously, we believe that's a really good thing, because if we were the only ones doing this, either we're way far ahead or there's something wrong with it. But we believe that other people will be jumping into this field, or have jumped in already. We just don't know, because the patents are not published for 18 months, and I mean, we started filing patents before 18 months. But it's for us. We're very thankful for our scientific advisors, people like Dr. Santosh Kesari at the John Wayne Cancer Institute, and numerous collaborators, because we have been able to start on such an exciting project, which we do not know anybody that's doing anything similar. So we, as the point is, in a very humble way, we do believe that this is a first-in-class therapy.
Yes, and you raised the point about others coming in. That's one of the things. When you have something significant, you have to expect that and do exactly what you just described in getting advisors and being the lead company in the field. So this sounds like it has many applications in a wide variety of diseases. Why did you pick stroke as the first indication?
I think we are very aggressive as a team, and the corporate culture of the parent company, and obviously the subsidiary, has been a very aggressive one from the beginning. And we just saw stroke as, obviously, people in the field, they call it a graveyard of biotech companies. And we just said, let's go for the most aggressive one just from the get-go, and see what we can do. In the animal models, we have seen very great therapeutic effect in animal models. The other thing with stroke that's very interesting that a lot of people may not know is that there's multiple studies that show patients after a stroke, patients who mobilize the stem cells. So this is actually a little bit counterintuitive to what I was presenting. And the reality is nobody really understands why, but there's multiple studies that show, if you look at 100 people with a stroke, people who have an increase in the stem cells in the circulation after the stroke, they always perform better. So what happens is, when you have a stroke, you have a big generation of molecules such as stromal-derived factor 1, which is a signal that injured cells make. So what is believed happens is people who have a stroke, they have this big influx, big generation of stromal-derived factor 1, SDF1, which acts as a beacon to call in the stem cells, from the bone marrow, to exit the bone marrow, go into circulation, and try to heal it. And this is different than a heart attack. So people who have a heart attack, there is increase in circulating stem cells, but from the literature that I've seen, nobody's really shown a black and white demonstration of correlation that more stem cells mobilizing in heart attack, endogenous mobilization, results with better prognosis. whereas in stroke, there's numerous studies showing this. These receptors for the SDF1 are also found on the cells of ImmCelz. So we believe that by giving the ImmCelz at the proper time, and the ImmCelz being modified cells of the person's immune system modified by being exposed to stem cells, we believe that we're going to be giving cells at the right time, and intravenously they will home and they will start a regenerative process similar to what we see in the animal studies. So we chose the model primarily. We chose the disease indication primarily because we had very nice animal efficacy data, and we also were able to see very potent in vitro signals that will correlate with the clinical situation. And for the last reason, because it has been one of the toughest indications for our stem cell companies. Stem cell companies have done some great work in it. Athersys, a lot has been learned from the work Athersys has done, and obviously from some of the animal studies that when you connectomize. So those studies showing that we know the spleen is an immune organ. When you give stem cells in stroke models, and you take the spleen away, then the efficacy of the stem cells in animal models goes away. So that was one of the indications that in stroke, at least, the immune system plays a role in some of the therapeutic effects of stem cells. What we're doing is we're basically recapitulating stem cells, but we're doing it through the T cells and through the immune cells. The other point that's supposed to be mentioned here is that we can give a lot more programmed immune cells to the patient than you can give stem cells. So when you give stem cells, the majority of stem cells get stuck in the lung. With stem cells intravenously, when I say stem cells, I mean mesenchymal. With mesenchymal stem cells, you have a limit to about 300 million. If you go more than 300 million, they all get stuck in the lung, and then you choke. Right? It inhibits your ability to take up oxygen. Because of the smaller size of the patient's immune cells, we can generate much larger doses that we can infuse back into the patient. And so that is something that we will be studying in future clinical trials. But right now, we chose stroke because of animal data and because it's a very tough indication.
Yes, certainly a tough indication, but large population, unmet medical need, and no effective therapies. So it's certainly one that any success at all will have clinical impact on the patients and bode well for the rest of the company and the technology. So what would you say is the strongest advantage to ImmCelz compared with other regenerative therapies?
Well, I would say on the one hand, because it's personalized, it seems to have more benefits in various animal models. When we have not presented the data, but we have done some back-to-back comparisons. So we think, on one hand, because it's personalized, is one of the advantages. The other advantage is because the immune system always finds its way back to homeostasis. So it's not one of those things where we're doing a permanent or a potentially permanent alteration. When you give an allogenic cell population, sometimes you don't know what it's going to do long term. Whereas this is just an autologous population that'll find its way back to homeostasis. And I think also the advantage is that with the immune cells, at least the T cell and B cell compartment, you have the ability to induce memory, immunological memory. Well, there's certain implications of this memory process where the memory process may be applicable to altering the long-term outcomes of other processes associated with health. So we think primarily it's because it's autologous, it's an allogenically influenced therapy gives an advantage. Some of the in vitro data makes us really excited about the potential advantages. And again, the size of the cells, the fact that we're using immune cells, I think is what makes it really exciting.
Great, yes, I agree with you. This is a very interesting technology and clearly differentiated from the other companies in the field. So I don't have any other questions, but I would like to thank you for that presentation. Very interesting technology, and look forward to following your clinical progress and the company as well.
Thank you for joining us for this C-Suite interview presentation brought to you by Channel Check. Visit our YouTube channel for more interview content, as well as virtual roadshows and conference presentation replays. New content is added regularly, so subscribe below to stay up to date. Visit channelcheck.com or click the link in the description to access equity research, news, and advanced market data on this and the 6,000 other small and microcap companies listed.